In this video, we're going to talk about some of the types of inheritance patterns we see that don't follow Mendel's law of dominance and recessive, independent assortment, and segregation. So Mendel worked with a very simple system. P's are genetically simple. Most traits are controlled by a single gene. Each gene only had two alleles. One was completely dominant to the other. And it made interpreting the results pretty simple. We talked about why that was helpful for Mendel. But the relationship between genotype and phenotype is usually not quite that simple. We have exceptions to the dominance rules. Incomplete dominance is one of those exceptions. In incomplete dominance, individuals that are heterozygous show an intermediate or a blended phenotype. It looks like blending inheritance is happening here. It's really not. An example here in the Japanese 5 o'clock flowers, you got three possible phenotypes from two alleles in one gene. You can see the genotypes and the phenotypes there at the bottom. So here's how it works when we do a typical Mendel cross. Start with true breeding red and true breeding white flowers and cross them. Instead of getting all red flowers, we got 100% pink flowers in the F1 generation. Allow them to self-pollinate. The red and the white genes come back, or the phenotypes come back, and you get a 1 to 2 to 1 ratio. With incomplete dominance, the genotypic and phenotypic ratios are always going to match up. And that's not the case with complete dominance or true dominance. ABO blood group in humans shows two exceptions, codominance and multiple allele trait. So in codominance, two alleles affect the phenotype equally but separately. It's not a mixture or a hybrid, but in the case of blood typing, the A gene causes an A protein to be produced on the red blood cells. The B gene causes a B protein to be produced. So if you have an A and a B gene, the A doesn't cover up the B doesn't cover up the A. It's not a hybrid mixture of an A-B protein, but instead you get both A and B proteins produced. Also in humans, we have three alleles for this one gene instead of just two. So it's a multiple allele trait. You have an A gene and a B gene and an I gene, which is for O blood. A and B are codominant to each other, produce both glycoproteins. I recessive to both A and B. So here's the inheritance. So if you have type A blood, you have two A genes, or an A gene and an O gene. You have type A antigens, type anti-B antibodies. They can donate to A and receive from A and O. B can have two B genes or B gene and an O gene. They have B antigens on their surface and anti-A antibodies. They can donate to B and they can receive from B and O. AB has an A and a B gene. They have both A and B antigens, but no antibodies. Because of that, they can receive blood from anybody, but they can only donate to AB. Type O has two O genes, so it doesn't have any antigens, neither the A nor the B. It has both antibodies. So they can donate to anybody, but they can only receive O. In pleiotropy, we see this in lots of genes where one gene affects more than one phenotypic character. Two examples of this in humans are achondroplasia, a form of dwarfism, and acromegalia, which causes gigantism. So here's Andre the Giant, a famous wrestler with gigantism. So let's look at achondroplasia. So let's look at two different crosses here. It's a dominant trait. In other words, you only need one copy of the dominant allele to display achondroplasia. So for most people, we're homozygous recessive for that trait. And that's an important thing. People tend to think that dominant traits are more common than recessive traits, and that's not always the case. So here's our other cross, two heterozygous individuals. So in the first one, we have one parent who has achondroplasia and one normal height parent. In the second one, we have two parents, both with achondroplasia. So let's look at our Punnett squares here and our results. So in this example here, where you have one normal and one achondroplasia parent, there's a 50-50 shot that the children will have dwarfism or be a normal height. So the ratio is 1 to 1. In the second one, you would expect to see a 3 to 1 ratio of dwarfism to normal height, or a 3 out of 4 chance of having a child with dwarfism each time. But instead, it's a little different because the homozygous dominant individuals, that's lethal. So the fetus is not going to survive. So what you actually see is a 2 to 1 ratio, or a 67% chance of having a child with dwarfism and a 33% chance that the child will have normal height. Epistasis is another exception where one gene covers up the effects of another gene. You see this a lot with color. You have one gene that causes pigment to be produced and another one that determines what the pigment will be. So in this case, you have to have at least one copy of the C gene to produce a pigment. If you have one copy of the dominant B gene, the big B gene, black color, 2 copies of the recessive brown gene with one dominant C gene, you get brown. But if you have 2 copies of the recessive allele for C, it doesn't matter what the B gene is because they're not going to produce pigment, they're going to be albino. So instead of getting a typical 9 to 3 to 3 to 1 ratio that we would expect from a dihybrid cross, the ratio is 9 to 3 to 4. Let's see how that happens there on our Punnett square. Here's another example in Labrador retrievers. There's an E gene and an O gene. Okay. The E determines pigment or no pigment. The pigment concentration is the B gene. So yellow labs have 2 copies of the small E gene. If they have 2 copies of the lowercase b gene, they also not only do they have yellow fur, but they have a brown nose, lips, and eye rims. If they have 1 copy of the black gene with the 2 no pigment genes, they get yellow fur with a black nose, eyes, and lips. Chocolate labs have at least one copy of the pigment gene, but 2 copies of the brown gene. They get brown fur, brown eyes, and so on. To have black lab, you have to have at least 1 copy of the big E and 1 copy of the big B gene. You see the different examples here. In polygenic inheritance, instead of having one gene with multiple alleles, you have multiple genes controlling one characteristic. So you get a continuous variation here. In humans, this includes things like skin color, height, weight, intelligence, and other behaviors. It's not you have just tall and short humans like you did in the pea plants. You have a wide variety of height, where most people are somewhere around the average, and then the numbers get smaller as you move to either extreme. You get a bell-shaped curve. But albinism, on the other hand, is a single gene trait. So you can have albino Caucasian or African American, or any person can be albino. So melanin is a brown color. So if you have 2 copies of the little a gene, you don't produce any melanin, and that causes you to have light-colored skin, light-colored hair, no pigment in the iris of the eye, and so on. Basically, there's an enzyme that converts tyrosine into melanin that these people are missing. Now, this creates problems because melanin is there to protect your skin from UV light.